Okay, so welcome everyone. Let's get started for today's Museum seminar. It's really my great pleasure to welcome Dr. Enfu Hui, also my friend and my collaborator, to come to a museum seminar and deliver the lecture for us today. So Enfu currently is associate professor in UCSD. And then Enfu actually complete his undergraduate study in Tsinghua University in biological science, and then further he completed his PhD training in biophysics in University of Wisconsin. So after he completed his PhD study, Enfu pursue his postdoc training briefly in University of Wisconsin, and then continue his postdoc training in UCSF. After that, he started his independent research program in UCSD from 2016, and then really published very productive, and then published a lot of great paper. And then currently 2021, he was promoted as associate professor in UCSD. So Enfu's lab is actually a member of Morris Cancer Center and the program of immunology in UCSD. His lab mainly works on cell biology mechanisms of immune checkpoints receptor through a unique combination of quantitative proteomics, cell-free reconstitution, live cell imaging, cell culture assay, and then mouse models. He actually is co-discovered member of cis interactions as a key mechanism in regulating immune checkpoint function. And then his lab showed that PD-L1 engage PD-1 in cis on the same cells to inhibit PD-1 function, which I think is really a brilliant discovery. Besides that, Enfu's lab also investigate whether and how additional immune checkpoints breaks operate in a conjugation with PD-1 to turn off T cell activity. And then they try to identify triggering molecules that turn these breaks on, and then signal transducers, which these breaks recruited to suppress the immune response. So I know that PD-1 and PD-1 is really the favorite molecule from Enfu's lab, but besides these work, Enfu's lab also demonstrate that PD-L1 interact with other co-stimulant molecules such as CD28 in the cis interaction manner to protect T cell immunity by simultaneously inhibiting both PD-1 and CTLA4 access. And his lab also dissected intracellular pathways of immune checkpoints. They recently showed that PD-1 and BTLA employ SHP2 and SHP1 respectively to inhibit T cell function. And then they also defined molecular features that underline the specificity, divergence of PD-1 and BTLA, and the mechanistic convergence of PD-1 and TIGIT. So all these findings are actually end up with high-profile publications, which potentially could lead to the development of novel biomarkers and drug targets of cancer immunotherapy. So with all these very productive and very groundbreaking discoveries, Enfu actually received numerous different awards and honors, which including serial scholarship, and then he was also nominated and then elected as a pure biomedical scholar. And also recently he got R37 Merit Award for early stage investigator from the NCI. So when actually Enfu told me he has recently have some really exciting discovery for the new cis interaction on the T cells, and I can't just wait to ask him to give us the lecture. So I think today he's going to talk about the CD28 and CTLA4 cis interaction on T cells. We're really excited to have you here. Thank you, Enfu, for delivering this lecture for us. Please go ahead.
Thank you so much, Tran, for this very thorough, accurate, well, I guess, really great introduction. It's a real pleasure to be here and share our new work with this wonderful community. Yeah, it's great that Tran gave a lot of introduction already, so I can just scan through the first few slides quickly. Okay, all right, so I guess, right, T cells are, per our body identify abnormal cells, and so the T cell response is requires its physical interaction with the target cells or antigen-presenting cells to form this synaptic-like structure. And at this interface, T cells receive various signals that dictate its functional response. And these signals can be divided into two types. The first type is antigen-specific signal, which is mediated by the T cell antigen receptor, or TCR, which bind to a short peptide antigen displaced by the MHC molecules on the target cells, or APC. But this axis is not sufficient for T cell activation. And the second signal through CD28 is also required. And the CD28, important CD28 is highlighted in many literatures. Actually, I just saw on Twitter that yesterday, 3/28 was named as T cell activation day. So that speaks to the importance of CD28. And CD28 interact with its B7 ligand in trans on the antigen-presenting cells to promote various functions of T cells. On the other hand, T cells also express inhibitory receptors to restrict their activities. One of them is CTLA4, which is thought to compete with CD28 for the same set of B7 ligands, CD80 and CD86. And CTLA4 is known to bind to these ligands with higher affinity. Therefore, the competition model. Another important inhibitory receptor is PD1, which bind to its own ligand PDL1 in trans and to deliver inhibitory signal to T cells. And this CTLA4 and PD1 are required to reduce the T cell activity and preventing autoimmunity. But on the other hand, they can also be hijacked by viruses and tumors to achieve escape immune surveillance. The blocking antibodies of CTLA4 and PD1 pathway has been approved by FDA for cancer immunotherapy, but these antibodies are not ideal. The response rate are quite low, and they always typically associate with autoimmune toxicity. And to reduce the toxicity and improve the benefit, we need a better understanding of these co-stimulatory, co-inhibitory molecules. As Tran mentioned at the beginning, my lab use a variety of approaches to study T cell co-receptor signaling with a focus on the immune checkpoint receptors. So we and others have contributed to the emerging concept of cis signaling. For example, we found that this PDL1 and CD80 or B7-1 can interact with each other in cis on the surface of antigen-presenting cells. And this was discovered by using a very nice FRET assay developed by my postdoc, Yunlong Zhao, who is really a brilliant scientist and supported by K99. Yunlong co-expressed PDL1 and B7 on this HEC293 cells surface, and then label PDL1 as B7 with donor and acceptor fluorophore respectively. If they interact in cis, then the PDL1 fluorescence would be transferred and quenched by the B7 fluorescence, at least partially. And then if we bleach the B7, we would detect an increase in the PDL1 fluorescence. And this is indeed what we saw when we bleached the B7 molecule. We can see an increase in the green fluorescence from PDL1. This allows us to calculate the threat. And this approach focuses on one cell at a time and eliminate the chance for trans interaction. So that's what we love about this microscopy-based single cell measurement. So this heterodimer could have a potential impact on the three co-stimulatory pathways. Since this is published paper, I just to tell you the conclusion here. The B7 can block PDL1, PD1 interaction in cis because if B7 and PD1 bind to the overlapping region on PDL1. However, the heterodimer is fully capable of activating CD28 in trans, just like the B7 monomer or homodimer. And surprisingly, however, the CTLA4 pathway was also diminished by this cis interaction, even though CTLA4 and CD28 compete for a similar area on B7. This turned out to be mediated by a avidity effect because in the absence of PDL1, B7 form homodimers that engage CTLA4 homodimers through a high avidity lattice. Whereas in the presence of PDL1, this lattice formation is disrupted. So this illustrates a striking example of a exclusive cis interacting pairs. And this pair only interact in cis, but not in trans. And this is demonstrated by independent assays from Gordon Freeman's lab and Yunlong's FRET assay. Okay, then we asked the reciprocal question. For those conventionally trans interacting pairs, can they interact in cis? And if so, what is the functional consequence? And this is a legitimate question because T cells not only express receptors, but also express various ligands. So they co-display receptor and ligand. So we focus on this B7 CD28 pathway. So this pathway is typically thought to occur in the lymph node or spleen, where there's a lot of professional APCs that display this B7 ligand on their surface so that it can activate the CD28 pathway in T cells in trans. However, in peripheral tissues, those tissue cells, non-immune cells, do not express B7. Okay, what makes professional APC professional is part of the reason is that they display B7, but those are not displayed by non-immune cells. And so this raised the question whether and how T cells receive CD28 co-stimulation in tissues. So we also know that B7 is displayed by T cells. So given the cis interaction we discovered for other pairs, we wonder whether B7 can engage CD28 in cis, and whether this cis interaction is productive and activate CD28. This may be important in tissues. So this has been a really wonderful collaboration with my long-term collaborator, Jack Buey at UCSD, and his staff scientist, Christine Calvin. And then Yayuan was is currently an undergrad student working with Yunlong. So this was led by Yunlong, and And this was also facilitated by Tran, who generally share with us the CD28 knockout mice that enable us to figure out the physiological relevance in vivo. Okay, Yunlong first decided to measure B7 induction in CD8 T cells. And this is OT1 splenocyte treated with peptide. You can see that the T cell OT1 cells induce B7 expression over time and remain B7 positive. And then Yunlong decided to use this his threat assay to determine whether this cis interaction happens. And indeed, he saw cis interactions. So in this case, it's CD28, CD80 threat, and this threat signal can be disrupted by abatacept, which is the CTLA4 Ig that bind to B7 with high affinity to block the CD28 binding. And this threat was also seen for CD28, CD86 pair. It's also blocked by abatacept. So what about the functional consequence? Does this cis interaction merely compete with trans interaction as a blocker, or is actually productive itself? So Yunlong developed this T cell reporter lines that couple NFKB transcription to GFP expression and AP1 to mCherry expression. And then he mixed these, co-culture this T cell reporter lines with APCs, a large excess of APC, 100-fold excess. And the reason we did that is to prevent the TT contact, to make sure that T cells can only undergo possible cis signaling, but no trans signaling, okay? So in the first condition on top, you can see that there's no antigen, no B7, very little signal for GFP and mCherry as expected. When we provided antigen only, we see a right shift of both signal, and this is a TCR response. In the third row, we then add trans B7 on top, and we see a further shift, right shift. This is due to trans B7 CD28 signaling as expected. In the fourth row, we move the trans B7 to the cis side, and we see a similar degree of signal for NFKB and AP1. And this is also true for IL-2 response. So this result suggests that the cis B7 CD28 interaction is productive. It activates CD28 and promotes T cell signaling. Going in vivo, we decided to first use a B7 knockout mice to eliminate this B7 expressing B7 expression on professional APCs, so we don't have trans signaling going on. Then we inoculate B16 ova melanoma tumor to this mice, and then primes OT1s in vitro, and to induce B7 expression and to enable cis signaling. We transfer these OT1 cells to the mice and measure the tumor growth with or without blockade of the cis interaction by abatacept. You can see that in the red curve, when we block the cis interaction, there was an increase in the tumor growth, suggesting impairment of the anti-tumor activity of the CD8 T cells. This was corroborated by this fewer T cells count in the tumor cells, in the tumor tissues, and fewer central memory T cells in tumors, and less cytokine production when the cis interaction was blocked. Histology analysis showed that the T cells are very sparsely distributed in tumors, and suggesting that they probably have to rely on the cell-intrinsic, cell-autonomous cis signaling to survive and maybe infiltrate to this type of tissues. So as biochemists, we wonder, how could this type of interaction, cis interaction happen for typically trans interacting pair? Structural modeling suggests that indeed the most stable binding form of these two proteins are the head-to-head mode. Okay, so how could cis head-to-head binding occur on the same membrane? We consider two possibilities: protein bending shown on the left, or membrane bending shown on the right, to enable head-to-head interaction. To distinguish these two possibilities, we consider the distance between their intracellular domains. On the left, they're fairly close. On the right, they're pointing in the opposite direction and are separated by two lipid bilayers plus the extracellular dimension of this complex. So this would be too far away for a thread to happen, whereas on the left, thread should be able to happen. So in this case, we just move this thread pair from the extracellular domain to the intracellular domain to distinguish these two models. Here's the result. I want to first show you the positive control on the right. This is the ligand-cis interaction between PD-L1 and CD80. You can see Fred in the intracellular labeling as well, suggesting that these two ligands bind to each other in a side-by-side fashion, probably shown as shown in the left scenario in the previous slide. However, for CD28 B7 cis interaction, they don't produce intracellular Fred, similar to this blocked condition, okay, and much lower than this positive control, and similar to this negative control, that non-interacting pair, CD28 and PD-L1. So these data support this membrane invagination model. Well, can we see it? To do so, Yunlong used this split apex system, in which he fused two halves of the apex enzyme to the two interacting proteins, CD28 and B7, shown as A and B here. When they interact, they would bring the two halves of apex together and reconstitute the functional enzyme. And then this allows apex to catalyze the polymerization of diaminobenzidine. And this can be then stained and then appear dark under electron microscopy. So basically, you can see the dark spot under EM as a readout for the cis complex. On the left is our positive control, the PD-L1 CD80 pair. These interactions you can see largely occur on flat membranes. However, for the CD28 CD80 cis pair, they are largely found in those membrane invaginate, confirming that membrane invagination model. All right, so what is a natural physiological process that would produce membrane negative curvatures or membrane invaginations? Endocytosis. And Yunlong was indeed able to make some connection between cis signaling and CD28 endocytosis. He engineers two types of CD28 mutant with differential endocytosis activity. One mutant has a loss of function in terms of endocytosis, shown in blue here. And this is similar to the negative control, which is done for wild type CD28 on ice, which inhibit the endocytosis. And the other mutant, shown in orange here, has a gain of function in terms of endocytosis. You can see that it internalizes much faster than the wild type CD28. This is also reflected in the size of the intracellular populations of this mutant. He then introduced these mutants to a CD28 knockout Jurkat T cell lines together with wild type, or individually, to 3 separate lines, and asked how they respond to cis ligand or trans ligand in producing IL-2. You can see that in the cis signaling format, the ability of this mutant to produce, to induce IL-2 production positively correlate with endocytosis. Whereas in the trans signaling format, it shows anti-correlation. And this makes sense for this gain-of-function mutant because it's largely intracellular, meaning that it has limited access to trans ligand. Perhaps more importantly is that we now identify a mutant, this blue mutant, this endocytosis deficient mutant that is specifically deficient in cis signaling, but perfectly normal in trans signaling. So this allows us to decouple the cis contribution from the trans contribution and test this in vivo. So we, again, so in this case, we use this mouse tumor model, but in this case, we use wild type mice so that they have the normal professional APCs that display B7, meaning that they have intact trans signaling. We want to ask, on top of this trans signaling, whether the cis signaling is still meaningful. So again, the same tumor model, but here we are using this CD28 knockout OT1 cells, reconstituted with either wild type CD28 or the mutant CD28 that lack cis signaling but not trans signaling, okay? And then we measure the tumor growth. You can see that this mutant CD28 expressing OT1 cells has a defect in controlling the tumor growth as compared to the wild type, demonstrating the importance of the cis signaling in supporting the anti-tumor activity of CD8 T cells. So we then wonder where these cis signaling happen in T cells, okay? And I show you some evidence that occur in negative curvatures, but where do we find those negative curvatures? For trans signaling, it has to happen at the immunological synapse because that's where the receptor and ligand find each other. But for cis signaling, in principle, it can occur in the synapse or outside of the synapse because they bind on the same cells. To answer this question, Yunlong did this confocal assay to visualize the CD28 distribution when the T cells form conjugate with a Raji cells that lack B7. We use this B7 deficient Raji cells to get rid of trans signaling. Okay, so in the first row is the control that doesn't have cis B7. You can see that the CD28 is homogeneously distributed along the T cells plasma membrane. No enrichment to the synapse, and it does not seem to recruit its downstream effector PKC theta. But when it provided the cis B7 ligand, you can see that the CD28 now polarized to the immune synapse and becomes very vesicular. You see this intracellular CD28, suggesting there's this coupling with endocytosis. And then this synaptic CD28 seems to now recruit the PKC theta. They're activated by the cis ligand. In contrast, this endocytosis deficient mutant failed to enrich to the synapse even in the presence of cis B7. And they did not recruit the PKC theta effector. They're not activated. All right, so you might wonder, what is really this endocytosis mutant of CD28? So this diagram shows some key signaling motif in the CD28 cytosolic domain. At the N terminals, there is a PI3K binding motif, this PYMNM motif, okay, that recruit PI3K and trigger endocytosis. In the C terminals, you have a PYAP motif that recruit both LCK and PKC theta. So this is known as the signaling motif. And so the CD28 endocytosis deficient mutant is basically have a phenylalanine to replace this phosphatidylate here that recruit PI3K. Therefore, it's endocytosis deficient. And Yun Long further made some interesting connections with the membrane remodeling protein called SYNAX9. And this is a very multifunctional proteins that contain a bar domain at the C terminus that act to bend the membrane into tubules. And next to it is a PX domain that allows SYNAX9 to interact with PI3K. And then it also has these low complexity domains that bind to AP2, clathrin, and the SH3 domain that bind to dynamine. So you can see that you can imagine this protein is involved in endocytosis and membrane remodeling. And this was a very beautiful eLife paper recently showing that the SYNAX9 is able to pull membrane tubules from the immunological synapse. And these very new tubules invaginating from the immunological synapse and extending into the T cells. And so this is what we think is happening. Perhaps CD28 bind to PI3K and PI3K bind to SYNAX9 and allow this type of tubulation. So Yun Long decided to visualize this. And we don't have this fancy EM in hand, so we decided to just use this confocal microscopy to take a look. The colocalization of SYNAX9 and lipid and potentially CD28. So these cells express both CD28 and the B7 ligand to allow for cis signaling, and this cells is then dropped on a CD3 antibody displaying lipid bilayer to activate the T cell receptor. You can see that both SYNAX9 and lipid seems to form clusters, okay? And this is consistent with the ability of SYNAX9 to induce the tubulation. This is probably what happened in the synapse. You have these tubules forms. And indeed, in the z-axis, you can see that there are SYNAX9 signals that colocalize with lipid signal and go inward into the cells for a long distance. And CD28 colocalize with SYNAX9 and the lipid at the immunological synapse. But it doesn't seem to go into the cells very much. It seems to stay as the tooth-like structure at the immune synapse. It seems that the cis signaling only happened at the synapse. And this is consistent with the confocal image that I showed you previously, that the PKC theta seems to be recruited to the synapse rather than the vesicles. Okay, and the mutant doesn't do that. The mutant failed to enrich to the synapse, much less than the wild type, and didn't form clusters very well. So this is what we're thinking as a model. The B7, we think B7 marks more functional CD8 T cells and promote various functionalities of CD8 T cell function. And they do so because the B7 ligands on T cells can activate CD28 in cis. And we think these synaptic invagination structures provided curvature to promote such cis interactions. And this is perhaps mediated by PI3K and SYNAX9, the membrane remodeling proteins. And the reason we think that CD28 doesn't go inward, go deeper, and stay in the synapse is perhaps that this tubular structure has the sharpest curvature at the synapse. It's like the vesicle neck that so narrow that allow the cis complex to go across the lumen of the tubule. But we need more study to demonstrate that. All right, so Now you might wonder how T cell get those B7 ligand. So let me tell you a separate story led by my very talented graduate student, Preston Dennett, Jibin Zhang, and super productive postdoc Xiaozheng Xu, now a group leader in Sanofi. And this was also a collaboration with Alice Sherrod at Yale University. So let me just tell you the simple conclusion that there are two sources of B7 ligand on T cells. And they can be endogenously expressed. They can also be acquired through a process called trogocytosis. And Preston decided to look through, study the mechanism of B7 acquisition by T cells. There are prior literature suggesting that CTLA4 as a B7 receptor can extract B7 from APCs. And so we decided to first look at CTLA4 using microscopy. So Preston used this CTLA4 expressing JERCAP line to conjugate with CD80 GFP expressing Raji cells. 1 hour later, he took this confocal image. He saw massive transfer of CD80 from the APC to the T cells. And the transfer of CD80 seems to partially and only partially colocalize with CTLA4 in intracellular vesicles. And other CD80 signal, especially at the periphery, cell cortex, do not seem to colocalize very well with CTLA4. There are CTLA4 negative patches, suggesting there's other mechanism for transfer. And under EM, Preston and Alice was able to see membrane fragment or tubule-like structures, projections, extending from the Raji cells that seems to be captured by the JERCAT cells. And so this is also consistent with the known mechanism of trogocytosis. It's basically a way for cells, for lymphocytes to acquire membrane fragment from APCs and redisplay them on their surface. So now we know that CTLA4 is not the only thing that mediates the transfer now based on Preston's imaging. So we decide to look for CD28 contribution, and we decide to move the time point earlier, immediately after we form the T-APC conjugate. Now you can see that the Raji cells does extend these CD80 projections to the T cells, and these projections seems to be largely captured by CD28, much less by CTLA4, suggesting that it is CD28, but not CTLA4 that mediate initial capture or acquisition of the CD80 fragment. We decide to measure this process more quantitatively using flow cytometry. Here we used human regulatory T cells because these T cells express very high levels of CTLA4. We don't want to downplay the CTLA4 contribution in this process. So we co-culture the Treg with Raji cells expressing either CD80 GFP or CD86 GFP, and in the absence of antigen, ipilimumab, which blocks CTLA4 B7 interaction, or CD28 blockade antibody. Okay, and then we quantify using flow cytometry the amount of B7 acquisition on the T cells immediately after the conjugation. I just want to bring your attention here, and this is the Treg cells without conjugation. You can see that it does not have GFP signal as expected. And right after conjugation, the Treg acquire a decent amount of GFP signal. This is CD80 or CD86, in this case CD80. And blockade of ipilimumab has no effect in the acquisition, but blockade of CD28 decreased the acquisition of CD80. And this is also true for CD86. So this experiment demonstrates that it is CD28, but perhaps less for CTLA4 that drive the acquisition of B7. And we also confirmed this using Jurkat cell lines using CD28 knockout line. Okay, when we knock out CD28 from Jurkat, it loses ability to acquire either CD80 or CD86. And interestingly, this acquisition process also allows allow MHC class 2 to be co-transferred to the T cells. And this doesn't depend on the presence of TCR MHC interaction, because it also happened in the absence of antigen. So this is a bystander transfer of MHC to the T cells. Now, what about CTLA4? CTLA4 is a very controversial receptor, even though at the beginning I draw the CTLA4 on the cell surface, is actually largely intracellular. 90 to 95% of CTLA4 exists in intracellular vesicles, as shown displayed in this model. However, CD28 is primarily cell surface localized. So you can imagine that during the initial contact with T cells and APCs, CTLA4 has limited access to the B7 in trans, but CD28 has plenty of opportunity to do that. So it makes sense that CD28 mediate acquisition of B7. But what does CTLA4 do? David Sensom and his colleagues proposed a very interesting trans-endocytosis model to explain the cell extrinsic function of CTLA4. In this model, the CTLA4 extract and internalize the B7 ligand from the APC and direct the lysosomal degradation of these ligands. So the net outcome of this is that the B7 on APCs are depleted, and that would inhibit the B7 CD28 trans-signaling to another T cells, the cell extrinsic model. But this also raised an interesting question: how is intracellular localized CTLA4 able to efficiently deplete the ligand in trans? And what about this transmembrane domain here? Is it just floating in the lumen, in the aqueous solution, or is it embedded in the membrane? So we now consider an alternative model in which the CTLA4 might be capturing the B7 directly on the T cell surface and undergo cis endocytosis and deplete the B7 from the T cell surface. To test this, Xiaozheng did this confocal assay to look at what happens on those trogocytosed B7 molecules on the Jurkat cells, and using CTLA4 and CD28 as a landmark. Okay, and you can see that at the right after the conjugation, the B7 primarily colocalized with CD28 as membrane fragment on the plasma membrane, much less with CTLA4. But over time, the CTLA4 vesicles capture those B7 signals. And this capturing can be partially blocked by ipilimumab, as expected. So this experiment suggests that the trogocytosed B7 can be depleted by CTLA4 in cis in the same cell. To further test this model, we decided to use this flow cytometry readout again. So we mixed the human T cells with Raji cells expressing CD80 to allow for trogocytosis. And then we purify these CD80 decorated Treg cells and then mix them with a large excess of filler cells. We also spin them down to the plate to form a monolayer to restrict their movement and to prevent TT contact, to prevent the transdepletion of CTLA4. And here's the data. You can see that the untreated Treg without conjugation already express a little bit of CD80 as reported. And right after conjugation, Treg acquire a large amount of CD80 from the APCs. But over time, these CD80 were depleted. And block the CTLA4 can almost fully recover the CD80 expression on the Treg. But this didn't happen for CD28 blockade. So this suggests that indeed it is CTLA4 that deplete those CD80, acquire CD80. And this seems to occur in cysts because with or without filler cell didn't seems to affect this pattern. This is without, on the top, these conditions are without filler cell, these are with filler cells. All right, so what is a functional relevance for all of this? You know, you see from Yulong's story that the B7 on the T cells are able to activate the CD28 in cysts and promote T cell functionality in peripheral tissues. On top of that, these trogocytosed B7 and MHC can also allow T cells to auto-stimulate each other. So T cell basically now can function like an APC and become independent from APC, and they can stimulate each other. And this is the data. When we precondition the T cells with B7 expressing Raji cells and then incubate them to produce IL-2, they produce much more IL-2 than T cells that are not preconditioned or preconditions with B7 negative Raji APCs. And importantly, CTLA4 can restrict this process because when we block CTLA4, you see a further increase in the IL-2 production from this T cell auto-stimulation. All right, so this is like a graphic summary for the two stories, or maybe three stories I presented today. So you can see a lot of things happen at the immune synapse. There are not only trans interactions, but also cis interactions. And the cis interactions can happen on both sides. On the APC side, there are cis interactions between the ligand and that can regulate the relative strengths of the co-stimulatory and co-inhibitory pathway. On the T cell side, the cis interaction can activate the CD28 to promote T cell functionality, both in tissues and also perhaps in the lymph node to promote T cell priming. And these B7 ligand can also be transferred across the synapse from the APC to the T cell side. Okay, but this process, this auto-stimulation mediated by the cis signaling can be potentially dangerous, even though we show that it promotes tumor immunity, but it can also, may also generate this autoimmunity. And CTLA4 has evolved to downregulate this process through cis endocytosis. And ipilimumab can inhibit the CTLA4 function to upregulate the B7 levels on the T cells to promote T cell autostimulation. We think that this might contribute to both the therapeutic benefit and the toxicity observed for ipilimumab and other CTLA4 antibodies. So this is just the type of take-home message focusing on the function side. I think there's really these studies show that there's the functionality, multifunction of these signaling proteins in the B7 family. The B7 ligand can do at least three things. On the APCs, it can activate CD28 in trans. It can also block PD-L1 in cis, although this only applies to CD80 or B7-1, but not B7-2. And when they get on T cells, they can activate CD28 in cis. And we show that this process is promoted by endocytosis because it produces the negative curvatures for this cis interaction. CD28 is also multifunctional. We all know that it is key co-stimulatory receptor that deliver second signal for T cell activation, but CD28 can also acquire these B7 from the APCs to make T cells become more like independent from APCs. And this process is restricted by CTLA4, mediated cis endocytosis. So we now propose CTLA4 as a T cell intrinsic B7 sink or timer to limit the amount and duration of co-stimulatory ligand display on T cells. Finally, I'd like to thank you for your attention and all my funding resources for their support. I'd like to take any questions.